Now I'm going to explain the methodologies used in Human Genome Project. Let me just write. Methodologies, very important for 2 marks, even for your competitive examinations also. Okay, methodologies that is used in sequencing of human genome, that is in the project, Human Genome Project. Okay, simple thing. Okay, so this is now human being, and we know that he has got 37 trillion cells, huge number of cells. Okay, so I'm going to take one cell from his body, like I'm isolating one cell, done. And now let me just put it in the Petri plate, not exactly Petri plate, I'm just making you understand like how exactly this project is done, okay, or was done. So this cell now transferred into the Petri plate, that is in vitro, that is outside the body of the organism, human being. We know that within the cell what is there? The chromatin is there, that is nothing but chromosomes, 46 chromosomes are there. Okay, so let me just take this cell out now and show you. First thing is, I want to extract this chromatin from the cell. Is it easy? Not that easy. The first thing is you need to remove this plasma membrane, cell membrane of the cell. Okay, I have removed it. What is there now? The nucleus is covering our chromatin. So I need to remove this nuclear membrane. Done. So I have removed it. Now I got a long thread. Is this long thread? That chromatin is pure in its purest form. Only DNA is there. No. What is there? You see many things. You see the proteins, correct? So nucleosomes will be there, even NHC will be there, scaffold proteins, everything will be there. So I need to remove it. So it's like this. I'm just removing everything. So this is called as purification of the DNA. Got it? Purification. So there will be so many methods to purify the DNA. That means removal of some associated things with the chromatin. Okay, so finally I got a pure DNA, that is in the purest form, the chromatin. Next is, what I have to do is, I have to treat this DNA with one enzyme, that is called as endonucleases. Endonucleases. Don't even worry about all this procedure, you will understand it in detail in biotechnology chapter, how exactly it is done. Okay, so and even I will not disclose each and everything. You may lose interest of biotechnology later. Okay, so what I do is I'll treat with the endonucleases, and what is the function of this endonuclease? They cut the DNA, so you get pieces, fragments. Here I got of the fragments after treating with the endonucleases like this. But one thing to remember that all these fragments are entirely different. Okay, because they are different segments of the DNA, correct? Okay, I have got only one segment, that is this, and this segment is here, this segment is here, like this. So I have got DNA fragments. Let me write DNA fragments after treating with endonucleases. Next is here I have got only one the DNA fragment. During the experiment I may lose it. Okay, so I cannot afford the same procedure again. It's expensive again. Okay, so need a lot of machines and even the manpower also. To avoid it, what I have to do is I need to produce multiple copies of the same DNA fragment. Got it? Okay, same, everything. What I do is I just put it in a machine that is called as a PCR, polymerase chain reaction. You will understand it later. Finally, I get lot of same DNA fragments. That is same, exactly same. The sequences will be same because it is nothing but amplification. It's like a Xerox machine. Okay, the PCR, it's like a Xerox machine. You get multiple copies of the same DNA fragment, and here I will pick one and I will store all these DNA fragments in a box. We will understand it later, and put it in the gene bank. Okay, I'll keep it in the gene bank. There is a bank where we can store the genes. Whenever I require, I just take it out like that. Okay, I have selected. I do the same procedure for all. Okay, so same thing. Then is now what I have to do is I need to use, I want to know what is exactly the function of this DNA segment. I don't know anything about it. I don't know what is the function of this, whether it is containing a gene or not. You have already understood that if you take the entire human genome, genes will be situated at regular intervals. You will not see like throughout the human genome genes are not present. Okay, some base pairs or some nitrogenous bases acts like a gene, some no. Okay, so that's what like coding and non-coding sequences. Coding in the sense these sequences will code for a specific mRNA like that. So here let me just understand whether this DNA fragment contains gene or not. Okay, what I have to do is I cannot put it back to human body to see its function because it's already fragmented. What I have to do is I have to take one DNA which is nothing but acts like a vector. You know what is the meaning of vector. Okay, so I need to select one vector. Here in biotechnology, vector is nothing but a chromosome. The chromosome which is circular in nature. Okay, so like this. This chromosome is normally seen in bacteria. We call it as plasmid, which is circular. What is the other speciality of this chromosome? That is nothing but it can replicate without the help of anyone. So it is actually the thing. Okay. Here, for this human genome project, they used a special vector. They built a special vector, that is artificial vector, which is called as artificial chromosome. Okay, so they built one vector which is called as artificial chromosome. Okay, and they linked this DNA fragment to this artificial chromosome. Okay, I am going to link this to this artificial chromosome. See, I have linked it. So when or while I am linking this my DNA fragment into this artificial chromosome, so some portion of this artificial chromosome has to be removed. You could see it, the small yellow portion is now removed, but it is replaced by this DNA fragment. Now this DNA fragment is called as foreign DNA. Okay, this is now artificial chromosome. That is nothing but not called as plasmid because I have not extracted the plasmid directly from the bacteria. It is constructed artificially in the laboratory. You will understand it how and everything later. Now what I have to do is I am going to transfer it into a one host organism. Okay, they have used two host organism in this experiment, that is a bacteria. Okay, so bacteria. Other one is yeast, fungus. Bacteria and yeast, both are single celled organism, and they transferred this. Okay, once I link this foreign DNA, that is my DNA, into this artificial chromosome, the entire DNA now, the chromosome, is called as recombinant DNA. Recombinant DNA means it is a type of chromosome which contains two types of DNA. One is artificial chromosome in this case, the other one is a foreign DNA. Got it? Recombinant DNA. Okay, now I need to transfer this recombinant DNA into the bacteria or yeast. Okay, so like this. Done. Now what I am going to do is like I will observe. So bacteria obviously multiply, and during the multiplication, see, this also now replicates because it is autonomously replicating chromosome. Now this DNA fragment, which is my DNA fragment, human DNA fragment, which is now going to produce the mRNA, correct? So you are going to get the mRNA. That's it. So you are going to get the mRNA within the bacterial cell because this gene is now expressed in this organism. Now this mRNA is expressed within the bacterial cell, or it could be same applicable to the yeast cell also. Okay, so I am going to transfer it into yeast cell now. The same thing is applicable now. In the yeast cell also, this DNA fragment will express and produce one mRNA within the yeast cell. Later you need to remove this mRNA from bacteria or yeast cell. That's it. So it is understood that mRNA is produced. So that means this DNA fragment contains gene. Yes, it is understood now. Same thing. So this is the thing. If it doesn't produce the mRNA, so it is understood that one of the fragment is not a gene, doesn't contain a gene. It is understood like that. This is now sequencing of understanding the position of the gene and function of the gene. Okay. Now let me tell you the two vectors. If I am using bacteria as host, if I am using bacteria as host, obviously I need to build one artificial chromosome which is kind of like compatible within the bacterial cell. Okay, it's like it will replicate within the bacterial cell. Then that type of vector, the artificial chromosome, is called as BAC. What is BAC? Bacterial artificial chromosome. Note this one. Very important. You get this question. What is BAC? Bacterial artificial chromosome, which is nothing but artificial chromosome which is built to link the foreign DNA, that is nothing but human DNA fragment, okay, where if bacteria is used as host. Then if it is yeast, then yak. What is yak now? It is understood. Yeast artificial chromosome. Write down full. Yeast artificial chromosome. Very important. Could be asked even NEET or board examination. Okay, note this point. Okay. Oh, and one more thing. In Human Genome Project, name the organisms which were used as host. Bacteria and yeast. Only two. Okay, so that's it. Now let me tell you one interesting thing. I think you people have heard about cDNA. What is this C? We have heard about aDNA, bDNA, cDNA, zDNA. In this cDNA, what is this cDNA? Okay, so we just write that. Now I have not explained the methodology. I have said the procedure. Okay, now I am going to write the methodology. Let me just write the something about cDNA. What is this cDNA? cDNA, C means complementary. Complementary DNA, which is now produced because of the result of reverse transcription. Please note this point, reverse transcription. Okay. And now let me just explain. So this is now bacteria. I have inserted BARC, bacterial artificial chromosome, which is now recombinant, which is containing my DNA, that is DNA fragment. Now suppose if this expresses now produces mRNA. So I get mRNA here. If the gene, if this DNA segment now produces one mRNA, then that methodology is called as expressed sequence tags. It is also called as EST. Very important for your competitive examination. EST. Express sequence tags is nothing but identifying the gene within the DNA fragment if it expresses as RNA. If the gene expresses in the form of RNA, that is nothing but mRNA in this case, then obviously it is called as express sequence tags. This is one method used in Human Genome Project. Hope you understood. Okay. So what kind of it? If RNA is produced within the host cell, then it is understood that it is EST method. It is EST method. So even you can applicable, this is applicable to yeast as well as bacteria. So both are applicable. Got it? So if this DNA fragment now produces the mRNA, then this gene is expressed. That means this is a gene in the DNA fragment. So that is EST, express sequence tags, identifying the gene if it expresses as the RNA within the host organism, that is EST. Okay. Now I have got this mRNA using this EST method. Okay. So what I have to do is I need to construct the real, because mRNA is single stranded. You cannot store it. You need to convert this mRNA into the real DNA. That means I am recreating the original DNA fragment. The DNA fragment is here. Got it? Now I cannot remove it. Instead of that, what I have to do is now I will remove this mRNA. mRNA is now removed. Okay, I'll put it in the Petri plate. Okay, now I'm going to put some ribonucleotides. I think you know about it. What's going to happen? So, if the ribonucleotides are complementary to the mRNA sequences, nitrogenous bases, so it's going to bind and make it the double stranded. Okay, but remember that if uracil is present, then we need one enzyme, that is DNA dependent DNA polymerase enzyme for proofreading. Okay, I think not required now. Okay, so we got the DNA. That means I have created double stranded DNA from the mRNA, and this mechanism is reverse transcription. Yes, reverse transcription. So I have constructed this DNA based on the complementarity, because I am adding the ribonucleotides. These ribonucleotides will bind to the single stranded mRNA based on the complementarity. So I have created one DNA based on the complementarity, that is called as cDNA. So this is now cDNA. Got it? cDNA is not a natural DNA. It is not present in our body. It has to be created outside the body. And then I have created this cDNA because now I got to know that okay, this cDNA is nothing but the gene. I have recreated the gene, and now I am going to put it back to the gene bank. Let me write. So I'll put it back to the gene bank, and whenever I want it, I can take it. So it is understood that gene bank contains a specific gene. Got it? They will not store some useless sequences. Only genes will be stored to understand it for future. Okay, better for the future. Okay, so that's the thing. Now let me just explain one more thing, that is sequence annotation. The second methodology. This is specific approach. Specific method. Very specific. You are going to get the mRNA and you are going to recreate the DNA, double stranded DNA from the reverse transcription method. So DNA is now done. That is nothing but gene. In this case, sequence annotation. What is this sequence annotation? SA, the second method. Here, let me just draw our genome. Okay. Here I am not following EST method. It is a blind approach. So it is understood that this is specific method, and in this case it is a blind approach. Blindly I am doing it. First thing is actually I will sequence the nitrogenous bases. Determine the sequence of the nitrogenous bases. Okay, how exactly the nitrogenous bases are arranged in my genome, which includes coding. So we don't know whether this sequence is a gene containing a DNA segment like coding strands or coding segment, and non-coding segments. The first thing is sequence the nitrogenous bases present in the human genome, which includes coding and non-coding sequences. Both are involved, everything is involved, and later you need to assign the function. Then you need to mark, okay, this coding segment is a gene, this non-coding segment is not a gene, like that. Okay, assign the function to the DNA segment, that is as genes. That is sequence annotation. It's very lengthy one, so I will not explain each and everything. It's enough. Just remember the definition of it. Okay. So that's all about the methodologies of HGP. Please note cDNA also, that comes in express sequence tags. Hope you understood this.